The purpose of this video is to provide a demonstration of how to use the pLogo website to generate a pLogo image. The first step is to paste or upload your foreground dataset, or the dataset you wish to visualize. If you choose to upload the dataset, click on the Choose File button and browse for the file on your computer. Otherwise, you can paste the foreground dataset directly into the text box. For the purpose of this demonstration, we will be using a dataset derived from a paper titled Mapping N-glycosylation Sites Across 7 Evolutionarily Distinct Species Reveals a Divergent Substrate Proteome Despite a Common Core Machinery, published in Molecular Cell in 2012 by Zielinska et al. Specifically, we will be using data from Table S2, which are high-confidence N-glycosylation sites, and we will use the Drosophila subset. After pasting the aligned sequences into the text box, you'll note several important features. First, at the bottom right, are the total number of sequences in the dataset. Initially, there were 2,229, and following processing, there were 2,213 remaining. To understand the exact sequences that were removed by pLogo preprocessing, click on the Foreground Preprocessing tab below. You'll note 5 sequences were removed due to being duplicates, and 11 sequences were removed due to having invalid characters. By clicking on these numbers, you can actually see the sequences that were removed. Additionally, to download and view the entire preprocessed dataset, click on the link below the Choose File button. Below this link, you'll notice a table which provides statistics on the high-frequency residues. In this case, asparagine at position 6 occurs at 100% frequency. By default, the pLogo tool will fix this position in the resulting visualization. Alternatively, you can click on the No Fix Positions radio button to change this option. The next step in generating a pLogo is selecting an appropriate background dataset. Because the foreground dataset in our example was protein-derived, we will click on the protein link. This reveals a wide variety of fully sequenced proteomes currently available on the pLogo web tool. Because our foreground dataset originated in Drosophila, we will click on the fruit fly link. You could also choose to upload your own background dataset or paste in a background dataset. At this point, you will note that both the foreground and background datasets have been validated, evidenced by green check marks below the Generate pLogo button. Clicking on the Generate pLogo button reveals the pLogo at the upper right, as well as a variety of pLogo statistics and parameters at left. The asparagine at the central position is located on a gray background because that position is fixed within the pLogo. We could also hover over other pLogo positions to get statistics on those positions. Clicking on a residue within the pLogo fixes that position and residue within the pLogo and generates a new pLogo in the context of those fixed positions. We could also click on the position again to unfix it. In this case, let's fix the serine at the +2 position. pLogos also have 2 horizontal red bars, which signify the 0.05 level of statistical significance. Residues within a pLogo are ordered according to their statistical significance, with the most statistically significant residues appearing closest to the x-axis, both on the overrepresented side as well as the underrepresented side. Let's click on the tyrosine at the -1 position. Finally, additional customizations to the pLogo can be accomplished by clicking on the Customize tab. This will be covered in a future video.